Okay, welcome back, guys. Once again, if you're here, you obviously don't fucking understand what we're doing in class. And it's something also learned in 7th grade and 9th grade biology, so you should still be fucking ashamed of yourself. But don't worry, I can fix your fucking problem, because I'm here to reteach the basics of meiosis. Leave a comment if you don't understand, and maybe I'll make a video explaining it later. As if I feel like it. Okay, so we're going to be doing the same thing again. Well, I'm going to just go through her slides, and yeah. Okay, then. What is above this? Nope, that's not really. Okay, so first of all, you'll need to know what fertilization is and meiosis. Meiosis is a process that we make 4 haploid cells, all that kind of crap. We make the daughter cells, which are used for sexual reproduction, and fertilization is when the 2 haploid cells come together. And yes, in a somatic cell, you need to know what a somatic cell is. It's a cell other than sperm and ovum. It's just basically everything we have. They have 46 chromosomes. And as you can see over here, chromosomes can be distinguished by their size, position on the centromere, and patterns of staining of certain dyes, blah blah. That's cool. A karyotype is those displays that you see sometimes of the 46 chromosomes, which shows 23 pairs of each chromosome, each paired with the same length, chromosome, position, staining pattern, blah blah blah. And then yeah, these homologous chromosome pairs carry genes that control the same inherited characteristics. And homologous, remember, homologous are not the same, so basically they have different alleles on them, so they look kind of similar, but they don't create the same effects. And here is those karyotypes. Note that they're pretty similar, they look pretty similar, but they have other things involved. So note there's 2, 4, 6, 8, 10, 12, 14, 16, 18, 20, 22, 24, 26, 28, 30, and then yeah, you'll get to 40. Blah. Cool. Yep. Yeah, okay, cool. They often use lymphocytes to get this information. So they get your blood, then they get your white blood cells, because lymphocytes are a type of white blood cell. Then they get the DNA from your white blood cells, and they do that. Cool. And yeah, fertilization. So basically, you make haploids when you undergo meiosis, and that means they only have 23 chromosomes, and you need to get back to that original 46 as a human. So when you bring 23 from the sperm and 23 from the egg, it becomes 46 again. Yes, and fertilization and meiosis alternate in sexual life cycles. Cool. Yeah, whatever. And yes, there are 3 types of life cycles, which are dependent on they kind of depend on which organism we're talking about. So the one we mostly do would be this first one over here. We're a type of animal. We basically, the only time we make the gametes is really when we need to make sperm and ovum. We don't stay in this stage for long. We very shortly staying there. Most of the time we're staying as a diploid. Multicellular organism. Yep. Then there's also another one where fungi and some algae like to be in. This would be the... What is it? This would actually be the least like ours, if you want to figure it that way. They spend most of their time in their... haploid stage. They spend most of their time in their haploid stage and spend only a little time in their diploid stage. They only go to their diploid stage when they need to... to restore their number of chromosomes. So they rarely stay their time there, but it's kind of similar. It's like the opposite of this one. Just think they spend most of their time. And then a lot of plants and some algae, they kind of have an equal amount. They spend a certain amount, they have this is actually called the alteration of generations. You might want to take note of that. It's called the alteration of generations. Basically, the first generation spends their time in their haploid stage, and that's called the what is it? It's called the gametophyte, if I'm correct. And then they spend their other half of the time in their sporophyte site, which would be their 2N, which would also be their zygote slash diploid. Yeah, cool. Okay then. Meiosis reduces chromosome number by copying the chromosomes once, but dividing twice. Yeah, so basically they have the same amount as if you had gone through mitosis, but instead they divide one time, and then they divide a second time, and now it's half. They've created half of what it originally had. That's the basic idea. Yep, and so the first division, they just break apart their homologous pairs. And remember, homologous pairs are not the same pair. So one of them was by your parent, by your dad. One of them was by your mom. Yeah. Yep. Although we should remember these crossover. We'll talk about that later. So, and then they break apart again in the second meiosis 2, and then they separate the sister chromatids. Nice. They have this beautiful picture actually that gives you an overview of everything you need to know about it. Okay. But this is a more zoomed in version, and let me just look. Okay, so this is more zoomed in. So yeah, prophase 1, cool. With prophase 1, it's they begin, what is it? They begin assembling. Note over here that they actually cross over, and they're connected by the chismata. This is important because different genes from the different homologous pairs will get ripped off and put onto each other, which will increase genetic diversity. And this state is called synapsis, and these connections themselves are called the synaptomeal complex. Okay, so yeah, basically the different not haploids. The different homologous chromosomes are attaching to each other, sharing their DNA, blah blah. Yeah. Okay, then we get to metaphase 2. They get lined on the metaphase plate, and the microtubulars are connecting to the kinetochore, kind of similar to what we were doing in mitosis. Then anaphase 1, the homologous pairs are now being torn apart from each other, basically. They get separated, and then telophase, they create 2 haploid cells. And they still consist of the 2 sister chromatids, but note that they aren't exactly sisters anymore, because the crossover caused them to share some of their DNA. And then we get to prophase 2. That's going to be basically the spindle apparatus starts forming again. And they begin moving towards the center, basically. And then you get to metaphase 2. They line up along the line. Yep, cool. They line it up. And note that they're not genetically identical. You see they have different portions on each other because of that crossover from earlier. Anaphase, they get torn apart again, basically. And then we have cytokinesis and telophase 2, and basically does the same thing. It makes 2 cells again. And now we have all the DNA we need to make an organism, half the DNA we need for the organism. And none of them are going to be exactly the same. Ah, yes. We're talking about those free events now that occurred during the first cell division. Okay, so during prophase, homologous chromosomes pair up and called synapsis. I was kind of talking about that. That was basically when they're like, the homologous pairs were joining together, and then there's this protein zipper thing called the somatomeal complex that holds them together tightly. Yep, so basically that's what they're doing. And the X-shaped regions that are formed are called the chemasta, and the sections of non-sister chromatid are exchanged. Basically, yeah, they cross over. One gets cut off and one gets put onto the other, and the other one that got cut off also gets put onto the other one. They just cross over, so to speak. And then note at the metaphase, it's not individual chromosomes are being aligned, like in mitosis. What's actually being aligned is the homologous pairs of the chromosomes. Yeah, so you would have 23 of them instead of 46 lining up. And then anaphase 1, yeah, it's the homologous chromosomes that are being torn apart, not single sister chromatids. Yes. The sister chromatids will be torn off in the second anaphase. Yeah, and the process during the second mitotic divisions are virtually identical to the ones in the first one. Yep, pretty much. Yep, so why do the sister chromatids stick together? Well, it's because you see the two of them have the sister chromatid cohesion thing that we talked about in mitosis, and that keeps them together through the first meiosis. And the cohesion itself is caused by these protein complexes called cohesions, which makes sense because cohesion, they're cohesion proteins, cohesions, duh. And then in mitosis, cohesions are cleaved at the end of the metaphase. Yep, that should make sense, because otherwise they would be permanently stuck together. Obviously, they need to be cleaved. If you don't understand that, goddamn. And then meiosis, cohesions are cleaved along the chromosome arms in anaphase, and at the centromeres in the anaphase. Yep, so basically, there's enzymes that come along and they cleave off their arms. And one very important thing to take note of. Note that mitosis creates two sister cells. Two identical. They're absolutely the same. But meiosis is very different. By the end of it, it makes four that are radically different from the original components. Okay, and this summary thing, which should be in your book, provides a pretty nice comparative analysis of mitosis and meiosis. The page number, let me look it up what it is in the book. Yes. It's 256 if you care to want to look at this more in detail. This gives a pretty good summary of the differences between the two. And now we can talk about the three mechanisms that create genetic variation. Okay, so the first one is basically stats. If you don't really understand what's going on, they're basically saying they line up in the middle and there's 23 of them. So, but they have a random chance of which side they'll be aligned onto. So think of a flip of a coin. Let's say heads is a blue one, and let's say tails is a red one. And if you get heads, whichever one you get first is going to line up on the left. So if you get heads, the blue one's going to line up, and if you get tails, the red one's going to line up on this side. And so since there's 23 of them, you would have to flip the coin 23 times. So there is 2 to the 23 possibilities in total, if you understand statistics. So basically, it'd be 1/2 times 1/2 times 1/2 times 1/2. Remember that kind of thing? Yeah, that's basically what it is. So 2 to the 23rd, which is about 8 million possibilities. Cool. But that's not the only form of genetic diversity. There's also crossing over. This is also very important. So basically, the crossing over causes certain of them to fragment off of each other and go onto the other one, and this creates recombinant chromosomes. Remember that, recombinant chromosomes. Recombinant just means recombined. So basically, you took two different things and they got combined together, recombinant chromosomes. And so they're different from their parents, that they inherited from their parents. And then, of course, we have to take into consideration fertilization. So, on its own, we had independent assortment, and that's 2 to the 23. But now remember that you have to get 2 to the 23 times 2 to the 23, because it's basically one sperm cell interacts with one egg cell. So you have to multiply them together to give yourself all those possibilities. And that creates 1 in 3 1 in 70 trillion possible combinations of chromosomes, and that's a ridiculous amount of variation. And that's ignoring crossing over. If we take into crossing over into consideration, it's pretty much maybe in the, I don't even know what's after trillions. I don't know. It's just a ridiculous amount. It's truly mind-blowing how many possibilities can create from these 3 things alone. Okay, yeah, so this is a diagram of them being separated apart. Yep. There's crossing over. Yep. And then, yeah. Cool. And then, why is this significant to evolution? Basically, all these genetic variations cause us to have different organisms that are slightly different from each other. And basically, evolution is survival of the fittest, remember? So basically, whichever ones have superior genetic variations will be more likely to create their offspring, so their genetic variation will become more prominent in the population. And remember that the genetic variations at the very first, come from mutations. Yes, if you would like more detail on this crap, you can just look in the book. I remember there was a lot more things while I was reading it that they talked about, but I'm pretty sure they're not that important, and yeah. Okay, so from this point on, what I'm going to be talking about is some very minor information that I neglected to talk about in my earlier video because I was too lazy. And that would be basically, what is it? The cell cycle regulation. Okay. Okay, so yeah, they have different checkpoints basically, and the checkpoints are to check to make sure that there's not anything going wrong in the cell. Because if there's multiple mutations or whatnot, it's bad for the cell, so there are checkpoints to make sure that there's not errors. And yes, there's a G1 checkpoint, and then if they get a go-ahead, so basically there's it's clear, and they'll go back. And then they have an S and G2 and M phases. They basically have tons of them. But if they don't get the go-ahead, they'll exit the cycle and switch into a non-dividing state called the G0 phase, or they have cellular apoptosis. It depends on whether the cell can be saved or not. So, yeah, here you go. Checkpoint, proceed. And then if it doesn't get the signal, go to G0, where the cell does not divide and is attempting to repair itself. And if it can't repair itself, cellular apoptosis may ensue. Cyclin levels rise throughout, sharply for interphase and fall abruptly. Peaks in the activity of one cyclin complex corresponds to peakins. Cool. So basically what they're trying to say is the cyclin CDK complexes increases, and it's a specific one called MPF. Cyclin concentration goes up as well. You see? Increase activity, increase cyclin, then decrease, and then increase, and then decrease, and then increase, and decrease, yeah. Okay, so basically what this is talking about the checkpoints, there's a CDK, and then there's MPF, and then cyclin is degenerated, and then degenerate cyclin gets thrown away, and then CDK goes back again, and it keeps going in this beautiful process. And it helps regulate the cell cycle. M-phase checkpoints ensure that all chromosomes properly attach to the spindle at the metaphase. Okay, so basically, now the M-phase checkpoint is just making sure that everything's been properly aligned, because if they don't get properly aligned, shit can end up really bad. You'll have extra or missing chromosomes. That's not a good sign. No, that will really wreck you. That's like how Down syndrome and whatnot happens. Okay, so protein signal to delay anaphase originates at the kinetochores that have not yet attached to spindle fibers. Yeah, basically, yeah. Okay, so basically, if they're not properly aligned, the signal tells it to delay itself, which then keeps the APC, which is the thing that activates anaphase, off. But once they're all attached, the APC activates, triggering the breakdown of cyclin and inactivation of proteins, uniting the sister chromatids together. So basically, the process can continue. We can continue down the path of the cell cycle. Okay, another important thing is the cell must have all the nutrients it needs, and growth factor usually also needs to be present. And then growth factors are basically things that can be produced by platelets and blood cells, by the tyrosine kinase receptors of fibroblasts, and types of connective tissue, blah blah blah. So basically, outside of the cell, there's different little protein complexes floating around, blah blah blah, and you need those to activate cellular division. If they're not present, they don't divide. Yay. Yes, in a living organism, platelets release PDGF in the vicinity of an injury. That would make sense, because you would want to start regenerating tissue that's been lost or damaged after an injury. So obviously a proliferation of the fibroblasts would help to heal the wound. Cool. So this is just basically a picture of it. Adding it, not adding it. Note that there's cells don't divide when there isn't, and then when you add it, oh my goodness, the cells proliferate like crazy. Yay. Yay. And then growth factors appear to be a key in density dependent inhibition of cell division. Yep. So basically, the growth factors, they get reduced if there's too much cells, blah blah blah. So a culture cell normally divides until they form a single layer on the inner surface of the cell culture. Yeah, that makes sense. If a gap is created, the cells will grow to fill the gap in. And then at high densities, the amount of growth factors and nutrients is insufficient to allow continued cell growth. So yeah, basically, there's not enough food and there's not enough growth factors to tell them to keep growing, so they just stop growing. Yay! That's it. It's pretty good, right? Because you don't want cells to just keep growing. That's bad. That's how cancer starts. And then most cells also exhibit an anchorage dependence for cell division. To divide, they must be anchored to the substratum. Typically, the extracellular matrix. Basically, what they're saying is the cells need to be connected to something usually. And this kind of is related to anatomy. This is actually quite related to anatomy. Basically, in a multicellular organism, the cell likes to have some sort of extracellular matrix. And the extracellular matrix, remember, were all those like little fibers outside and whatnot that they are connected to. Yeah. Yep. And cancer cells are free of both density-dependent inhibition and anchorage dependence. Yeah, basically, something went wrong in them and they just ignore. They don't need those. Those things don't inhibit them anymore. And they just keep growing and growing and growing and growing. Like over here. And abnormal behavior of cancer cells begins with a single cell and a tissue undergoes a transformation that converts it from a normal cell to a cancer cell. So basically, genetic mutation, and that genetic mutation just happens to increase your cells so that it doesn't have any inhibitions. It just keeps growing. Normally, the immune system recognizes and destroys these transformed cells before they become a problem. But if they can avoid being destroyed, they become a tumor, which are bad. Tumors are big mass of normal cells. And then if they stay at the original site in a clump, it's kind of okay. They're called a benign tumor, and benign means like not damaging, basically. So yeah, they create a benign tumor. But if they cause they don't usually cause serious problems, so it's usually okay, but it depends on where they are. Like, if you have a benign tumor in your brain, that's actually very bad, because it'll be pushing your neurons and whatnot. It'll be pushing your brain against your skull, and that's actually very bad. But in certain areas, like let's say in your leg, it's like they had to cut really deep and all that kind of stuff. It's like it's not really worth it because most likely they won't do anything.
你们还是昨天从那个烧饭做到，我睡觉还没做好。
Yeah, there's a lot.
也这种现在磨的处理好啊。
I mean, but I didn't do it straight either. Okay.
你可以买这个吗？
No, I don't have that much math for okay. Anyways, you just leave, and then a malignant tumor. Okay, ignore that. That was my dad, basically. Yeah, he was talking about my math. It's like, oh, it took too much time, blah, 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 blah. Okay, okay. Ignoring that. Malignant tumors are basically the bad kind of tumor. They're the tumor that likes to invade other regions. And then they can affect more than one organ at the same time. And this is a type of cancer we usually think of. They get detached from their original site, and then they either go into the bloodstream or they go into the lymphatic system, and they metastasize. The bloodstream is usually harder for them to go through because the rapid speed of them makes it hard for them to deposit. And then the lymph system has the disadvantage of the fact that the lymph system has where all your white blood cells are there, so your lymphocytes usually can capture most of them and kill them, but eventually they might get overwhelmed or they might miss one or two, and then it can metastasize. And this is basically a picture. It started out in just the mammary gland, then it spread out, it begins invading neighboring tissues, and then eventually gets the blood and lymph vessels, and it can spread everywhere. When you get to metastasis, that's pretty bad. Your chances of living dramatically go down if it metastasizes. Yep, and the treatment for metastasizing cancers is high energy radiation and chemotherapy or toxic drugs. Basically, radiation therapy is they get gamma radiation or some other high radiation, maybe it's X-ray, something like that, something that destroys cells. They point out where that is, and then it kills everything there, including healthy cells as well. That's pretty bad. Chemotherapy is basically you take a drug, the drug is extremely toxic. It's toxic to you as well, but it's more toxic to the cancer cells, so then it kills them off. Yay. And people are beginning to understand how a normal cell become a cancer cell. Basically, what they realize is during the cellular transformation, usually is almost always a genetic mutation that changes the cell cycle control system. So basically, the cell cycle control system doesn't inhibit the cell when it should, so the cell doesn't repair itself. And yeah, here, cell cycles are controlled, escape from the cell cycle controls. So basically, cancer cells divide excessively and they invade each other tissues because they're free of the body's controlled mechanisms. And basically, cancer doesn't stop when there's no growth factors because either they make their own or they have an abnormality in the signal pathway, so they can just ignore the control mechanisms, or they have a problem, the cell cycle control system, yeah. And cancer cells may divide indefinitely if they have a continuous supply of nutrients. Basically, the point is cancer is kind of a miracle. Cancer is immortal. It can't be stopped. It just keeps going and going and going. They'll just keep dividing till the end of time. Like, look right here. They're talking about remember that article we read earlier from the tumor removed from a woman, that Henrietta Lacks? They're saying it's still creating new cells. 50 years. 60-something years since it's been cut out of her. That's amazing. The cancer is pretty much immortal. Okay, and then we were talking about cervical cancer, HPV, and the pap smear. Yeah, okay, let's talk about this crap. Oh, never mind. Doesn't have much detail. Okay, if you really want to know what this is, basically, HPV is something that, if I'm correct, it inflames the area and it causes changes in your cells that mess with your cell signaling, and that increases your chance of cancer. Cervical cancer is basically cancer of the cervix. The cervix is the entrance to the uterus, remember that. And the pap smear is basically they put something in and it takes a small collection of cells from the cervix. And then it checks for abnormalities. They then check for abnormalities. If they find abnormalities, then they know you have cancer, and then they can go in and aggressively deal with it. Yeah. Okay, guys, congratulations. Now you know meiosis and cell cycle regulation. Okay, leave a comment below if you need extra detail, blah blah. Same old, same old. Cool. Alright, bye.